Good evening, everyone. I'm actually a new recruit to the Chinese University of Hong Kong last year, so I have only 1 year of experience to tell you about how I actually prepare for my proposal. But I really agree with the previous two speakers that I really think a lot of the points that they have explained to us are very important. And I have also got some experience and also opportunity to ask my peers about how they prepare for their proposal. So in this presentation, because I have only 1 year of experience, so I will tell you how I formulate my proposal. So I have submitted my proposal with the title here, Driving Pancreatic Beta Cell Regeneration by VEGF Modified mRNA. I have actually been trained as a heart person, so this is actually something new to me. But I still think that as a new PI, I should develop something different from what I have been doing in the past. So I was a little bit brave to propose a project that I do not have any track record, to be honest. But I do have the track record or experience in using the new technology, which is making the modified mRNA. And I submitted this proposal to the research field, regenerative medicine, and also diabetes and metabolism, because I'm focusing on pancreatic beta cell regeneration, which might have the hope to treat diabetic patients. So I think the choice of the field, I was told that this is very important because that would make sure reviewers understand what I'm going to propose. And the other things which is very similar to a lot of other proposals, which include a background, why I wanted to do this study, and generate a hypothesis and identify an unanswered question in the field, and support with preliminary data and also very clear aims of my proposal. So I think the most important point, as also pointed out by the previous two speakers, including the originality, novelty, impact, feasibility, and also collaborations. And I think collaborations is very important. As I said, I have not got any track record in this field. That's why I really have proposed 2 collaborators in the beta cell biology theme to strengthen the possibility that I can impress the reviewers that I have the ability to finish this project. So, in order to identify unanswered questions, I actually look at a lot of the papers and see that there are clinical trials using, for example, a pro-angiogenic factor, which I propose to use to induce blood vessel formation. But there are lots of clinical trials already showing that there are different methodologies. But we know that there are lots of side effects or the pitfalls why their approach fail. That's how I actually formulate my proposal. I was thinking like maybe we just target the same question using new technology. So I have also done some preliminary experiments showing that the old or published technology is not going to work. They cause side effects compared to the normal control. So I actually think that I can make use of what I have shown in another organ system, although it is published in the hearts, but I can use the same technology now in a new model system. So my proposal was actually focusing on new technology to solve a problem which I don't have a lot of expertise in, but I think that the crucial thing is the novelty and also the impact, because we have shown that previously it worked in the hearts, but now what about if we want to do the same thing in the pancreas? And I also emphasize a lot in the impact, because I think that in the medicine field especially, I think they want to see that whether my proposal can be translatable. So I have patented technology actually, and also we have worked with AstraZeneca and also Moderna Therapeutics to show that this technology can actually be used for drug development. So then I actually also point this out in my proposal to say that my work will be translatable. So, and then I also read the literature, and I try to identify a question that has not been resolved. Just like there is a question in 2001 published in Science that whether blood vessels can actually stimulate pancreatic beta cell to function. And I think this is a long-standing question, but no one actually has really know how to answer this. So I think I have formulated new questions and also trying to use new technology to solve the problem. So this is just to show that I have also include a lot of the preliminary data from other people. So in addition to my work, as I said, because I'm new to this field, I also need to show to the reviewers that I have read a lot of the papers in this field, and also use some of the preliminary data from other reports. Of course, I also have my own preliminary data in addition to the preliminary data from other reports. So I have, after using of my own track record in terms of the technology, and also doing a similar thing now is not in the hearts, but I also wanted to study something new, which is the pancreatic beta cell regeneration. So I formulate a proposal, which I have, I tried to make use of my expertise. And you also try to explain the model has some similar, have been supported by other papers, although it is a new model when I replace the new gene in the mouse model. And also I show very visualized results because I wanted to show to the reviewer that now I can actually induce more blood vessels in the pancreas. In the past, I did show it in the hearts, but I have never shown in the pancreas. So I actually show this in my proposal. It is very understandable by the reviewers, I think, because you can see by eyes that now we have more blood vessels. So I also incorporate a diagram in the end to just try to summarize all of my aims in the proposal. So I think this is quite important to just, because sometimes it's quite boring to read a lot of sentences. Now I actually include all the aims as a figure. So I think that is quite, that would help the reviewers to understand our central hypothesis. So, and I also show that, as I said, I don't have a lot of track record in the beta cell biology field. So I collaborate with local collaborator and also my colleague when I did my postdoctoral training in Harvard. They are the beta cell biologists. And then I also collaborate with bioinformatics in my department in order to show to the reviewers that although I don't have track record in beta cell biology, but with collaborations, it is possible for us to finish the project. So I think what I have included in the proposal include originality to identify unanswered questions, the novelty to utilize new technology, the impact because we are targeting more understanding for disease such as diabetes in my proposal, and I also need to show the feasibility in terms of my track record to be able to use the new technology and also high expertise in the hearts, and also to collaborate with beta cell specialists to strengthen the feasibility of my project. So I think this is the end, and thank you very much.